So you have to watch some really old television to get this one. It says single cell sitcoms and they're singing, and that's the way we became the bacteria bunch. That's the way we became the bacteria bunch. It took me a while to, I had to get the song back from the Brady Bunch. Cells, right? Microscopes allow you to see things expanded enough, magnified enough, that you can see cells. And cells wind up being somewhat important with some of the stuff we're going to talk about. We don't get into cells so much as in my other classes, but still going to talk about them a little bit. They are chambers. They're chambers with a mostly waterproof casing, with sometimes a casing around that. And it allows the chemistry, which is water and a bunch of stuff, inside the cell to be somewhat independent of the juice around the cell. You always need juice around an active cell because that's the only way to get stuff in and out. But the juice inside the cell can be significantly different from the juice outside the cell. And that's because it's got this semi-waterproof casing around it. Now, life falls into a couple of categories. Now, technically, single-cell living things are unicellular. I almost never use that term when I'm talking because I think single-cell comes across better, you understand it better. But if you're reading, you may run into that as a term, and they mean the same thing. Same thing here, although it's pretty much obvious that multi-celled and multicellular mean the same thing. So living things can be, their whole organism can be a single cell, or the whole organism can be more than one cell. And you would think, okay, that's it, that's either or. Turns out there's a gray area in between. Now, colonial applies to more things than just the unicellulars. And I'm going to ask you about this, so you want to know this term. A colonial species is a species that, when you find them in nature, there's a whole bunch of them clustered together, and they're doing, individuals within the group are doing different jobs. They're working as a unit where they split up some of the tasks. People are colonial, right? Under natural conditions, you find people in groups, everybody in the group's not doing the same thing. Things that travel in herds, like gazelles. Gazelles are not really colonial. You do find them in groups, but they're not, nobody's doing anything special to support the group. They may do some different things, but colonial specifically, the members of the group are doing jobs that help the group as a whole. And there are plants that are colonial, there are single-celled, this is single-celled organisms that are colonial. And so it's what makes them single-celled. You find them in clusters. If you split the clusters up, each individual cell can live on its own. Doesn't like to, tends to find other cells and get together and form another colony, but they can. We can't do that with you, right? We split you up into individual cells, they're all gonna die. And most multi-celled systems, you can't split them up and have the cells live on their own. I think about this, people in theory, if you put somebody out in the wilderness, in theory they could live on their own, doing all the jobs they would need to do to keep themselves alive. But people work much better in groups, splitting those jobs up. So these are the terms that apply to living things. This we've already covered. There's two major different types of cells. There's the bacterial cells, the prokaryotes, little, don't have special separate internal chambers. There's a bunch of chemical differences between them, which is good because a lot of prokaryotes can make you sick. And the good thing about developing antibiotics for bacterial diseases is we can mess with the bacterial chemistry and not mess up our own chemistry doing it, because bacteria do some things that we don't do. We're eukaryotes, bigger cells, chambers inside, specialized, chemistry a lot different. We're not getting into that. There's differences between animal cells and plant cells. We're not going to get into those particularly, but I do want to cover the original cell theory developed going on 200 years, 1838, so a few more years from now we'll hit the 200-year part. Two guys, Schleiden and Schwann, one of them working, looking through microscopes at animal cells, one of them looking through microscopes at plant cells, kind of got together, kind of pooled their knowledge and went, wow, there's some general rules we can come up with that just apply to life in general on the basis of cells. And they came up with 4 rules. And the rules have stayed pretty stable. I mean, we added a little bit to it, and you'll see that when I put these down. But they did a pretty good job of coming up with rules here. They decided that everything that could be considered alive is cellular. It's made up of at least one cell. Now, when you get a bunch of biologists in a room and you go, viruses, alive or not alive, viruses are not necessarily cellular, so they break this rule, and to biologists that are very rules-oriented, they go, well, viruses aren't alive because they're not cellular. There's a couple of other weird rules that viruses break that usually is on the list of things that living things need to be alive. But you know, the viruses don't care what we call them. Everything alive is at least one cell. So you look at these cells. Remember, they were looking at very tiny things. You could see stuff inside them moving around. They decided that things that were inside the cell, even if they were mobile and they moved around and stuff, they themselves were not going to be considered alive. So this is basically a definitional. This is a labeling thing, right? We love to label stuff. And they said anything smaller than a cell we're not going to consider that to be alive. We're going to consider it to be part of something that's alive, but not in and of itself alive. So they went, okay, no spontaneous generation. Cells only come from other cells. Now the blue thing was added later. Through 20 and 30, 40 years after Schleiden and Schwann, as people started to find that when folks were sick, that you could scarf cells off of them that were not like their body cells. They were getting bacteria. For a while, the theory of what was going on was that when people got sick, their own cells started to generate these bacterial cells, is that our cells would make bacterial cells. And it took a while for people to realize that no, bacterial cells can actually get from person to person. They're making the disease happen. That's the germ theory. But this was before the germ theory. Later, this got added. You only get bacterial cells from other bacterial cells. You only get eukaryote cells from other eukaryote cells. Cells produce cells that are like them. So the related was put in there, which, as an add-on, not a big difference. I honestly think that this is two guys doing research in the same situation that people that work in science now face. When you work in science, you invariably have relatives and people that you know. They go, so what do you work on? And then you tell them, then their eyes glaze over. And they go, so what can you do with that? You know, how is what you're doing useful? And so you come up with stuff. As you read it in scientific papers and press releases all the time, they always end with, we're gonna be able to do all this wonderful stuff. Most of the time, they're not, but it's a way to explain to relatives why your stuff is important, and also explain to people who may be gonna give you money for your research why it's potentially important. But, so they were able to go, well, we look at cells under the microscope, but cells are all pretty much alike. They're way more alike than they are different. So the stuff that I discover working with an amoeba is something we're gonna be able to apply to our own cells at some point. And some of that's true. Things that amoebas do, our white blood cells also do. So it's sort of kind of true. Scientists do research into basic stuff and then expand it to bigger stuff. But this is, if you took a bunch of people who work on cells and you go, give me a column of things where cells are alike and a column where cells are different, and make the different column longer, you can make the different column longer. There's lots of similarities, there's lots of differences, but this is part of the rule, because they wanted it part of the rule. It's not completely wrong, it's just a little iffy. So this is the size chart. Every jump is a 10-fold increase in size. So you got atoms way down on the bottom, and here's us, 10 meters, there's 1 meter, and then a 1/10 of a meter, and 100th, and 1,000th, and 100,000th, and This yellow range is the range where you tend to find actual living cells. And you can see, electron microscope can see in this range, light microscope can see in this range, the eye can see in this range. There's not a lot that's smaller, and there's not a lot that's bigger. Okay, why are there no cells that are smaller? Well, because cells have to do stuff. There are big bags of chemistry, and you gotta have so much in there in order to actually do the stuff that living things need to do. These little bacteria live in other cells and have the cell around them do a lot of the stuff that they would normally do. So that's the only way they can survive being small, is they gotta leach off the small cellular parasites. They live in other cells, and so that way they don't have to do absolutely everything that a normal cell would have to do. But the question always has been, well, why aren't cells bigger? Why don't we have big cells? We have big organisms, big living things, why not big cells? And I'll give you the standard answer, which is pretty much wrong. But you read it in a book, it always gives you this. And then I'll give you what's more likely the right answer. The standard answer uses math. You have a cell. How's it do stuff? Well, stuff needs to go in and out across the outside of the cell. If you double the volume of the cell, this surface doesn't double. You have to actually increase the volume by 8 times in order for the surface to double in size, what they call a cube root effect. So as cells get bigger, they're doing more stuff, but the outside, their ability to get stuff in and out of the cell doesn't keep up. So as they get bigger and bigger, they're more and more restricted in what they can do. That sounds perfectly reasonable, right? Here's the problem. That works if the cells are perfect little balls. Most cells are not perfect little balls. Your skin cells are very, very flat. If you double the size of a skin cell, you come pretty close to doubling the size of the surface, because they're very, very flat. And some cells have projections. There's all sorts of ways to fiddle with the surface so that you can kind of keep the surface in line with the size of the cell. So in the real world, that math answer, it sounds satisfying, but it doesn't really apply. I've also heard that, the cell can't run something that's bigger, but that doesn't really make any sense. Here's the more likely answer. As organisms got bigger, you had cells getting bigger, and then you had cells becoming colonial. A cluster of cells where each cell can do its own chemistry, contributing to the colony, works a hell of a lot better than a single cell trying to do all that on its own. Is that single cells, big, can't compete with colonial cells, eventually with multi-celled systems. So at some point, probably in the past, that was the way things were going. Big cells versus clusters of cells. Well, the big cells just couldn't keep up. And there are things that single cells do very well, so there's way more single cells than there are multi-celled organisms. In fact, if you put all the single-celled organisms on one side of a balance and all the multi-cells are on this side, so all the bacteria and amoebas and stuff over here, and all the elephants and the sequoia trees and the people and the horses and the plants there, these would still outweigh us. All the bacteria would still outweigh us. So there's not only a lot more of them, they form much more of what they call the biomass of the planet. So they're doing pretty well. They just don't do well in big sizes. And that's almost certainly the reason why you don't find giant cells. I mean, there's some places, if you look real carefully, you can find giant cells. There's amoebas at the deep, deep, deep bottom of the Pacific Ocean that you would be able to see pretty easily without a microscope. They're pretty big. But it's biology. Any rule you come up with, some crazy living thing somewhere is going to break it. Just a couple of terms to throw out there, because you may run into them doing some of the reading. You have the cell membrane, that's the barrier around the outside. Plants and fungal cells and some protists have a cell wall around that. Some of them have casings that kind of take the place of a cell wall. Our cells don't have a cell wall, we just have a membrane around them. Eukaryote cells have a nucleus where the DNA is processed, but lots of other bits and pieces inside as well. And there's juice in there where things can move around. That's called cytoplasm, cell jelly in Latin. And there's what they call organelles. Not really organs, but kind of sort of work like organs inside your cells. We're not getting into all of that stuff, except a little bit when we talk about the development of eukaryote cells, where we talk about where cells and stuff came from. So a lot of the detail stuff that we would be covering in very extensively in my other classes, we're not doing so much in here. So that is it for chapter 4, section 1.